Hello, welcome to another video by Mahmoud Nower. Welcome to Nower Yudarris. Today we'll be going through some biochemistry. We'll be going through one of the most important topics that you really need to know on the MCAT, and which is the amino acids. So we'll be speaking about 20 amino acids today. We'll be speaking about peptide bonds, protein structure, what are the steps to it, etc., etc. So stay tuned and let me know in the comments if you have anything to say, any topics you want me to cover, and let's start. So what's an amino acid? An amino acid contains an amino group. So that an amino group is basically NH2 and a carboxyl group, COOH. So I just recently made an episode on nomenclature for organic chemistry, so go check that out. So amino group comes from amine including nitrogen. Carboxyl group comes from carboxylic acid, but here it's not really carboxylic acid. So when I discussed it, it's basically OH and double bonded to oxygen. That's what a carboxyl group is. And this is an amino acid, NH2, a carboxyl group. And their properties are determined by the side chains they have. Now, amino acids have A alpha carbon, which is the chiral carbon on all amino acids, in which they have S configuration and L configuration and are optically active. Alright, so the alpha carbon is a chiral carbon. Chiral means center carbon, like the most carbon connected to a lot of important chains or groups. And then on all amino acids except for two. So two, you need to remember them that defy these rules. So you got the glycine, which is achiral, so it's not chiral. Glycine is achiral, and cysteine is an R configuration. So all amino acids are S configuration except for cysteine, it's an R configuration, and all amino acids are chiral except for glycine, it's achiral. So now we need to divide the amino acids into multiple groups. They're divided into multiple groups. Each group has its own properties, and which makes it unique. So we're going to start with the nonpolar and non-aromatic amino acids. So these amino acids don't have a benzene ring and they're nonpolar. So these include glycine, so the chiral, the one we spoke about, glycine, alanine, hydrophobic, valine, hydrophobic, and then proline and leucine, and you got methionine, has a sulfur group, and cyclic, and isoleucine, hydrophobic. They're almost all of them are hydrophobic, and nonpolar. Okay, so remember with me. So glycine, alanine, valine, proline, leucine, and methionine, isoleucine. All right, this is how you remember them. They're all hydrophobic. Okay, the way I look at it, or it's easier for me to remember, is that how short these amino acid names are in comparison to the ones we're gonna look at later. So maybe that's a good way, or just try to remember glycine, alanine, valine, proline, methionine, isoleucine, leucine. Also, the fact that there's two leucines here, leucine and isoleucine, kind of helps. Next, we have the aromatic. It's uncharged benzene ring. So like this group, they have a benzene ring on the side. So here you got three. You got tryptophan. Alright, it has double rings, not one but double rings. Next you have phenylalanine, it's hydrophobic. Okay, remember here, alanine, hydrophobic, phenylalanine, it's hydrophobic, and tyrosine. So you got here tyrosine, it's basically polar. If you want to look at it here, tyrosine, why is it polar? It's polar because of the OH group here, the alcohol group. So this is why tyrosine is polar in comparison to phenylalanine, which is, we could say it's alanine but with a aromatic or a benzene structure. So here, phenylalanine, they're hydrophobic, okay? And then tryptophan. So tryptophan here has two benzene rings. So basically the ones with the benzene rings and hydrophobic are three. Alright, so tryptophan, tyrosine, and phenylalanine, but tyrosine is polar, but it's hydrophobic. Okay, polar side chains, and they're non-aromatic, so they don't have what do you call it? They don't have any benzene rings. They're non-aromatic, but they are polar. So you got here four, okay, and you can look at them here. They're polar uncharged side chains, no benzene ring. So you got serine, you got asparagine, you got glutamine, and cysteine. Remember cysteine? Cysteine R configuration. Cysteine is the unique one, has an R configuration. It's a special case. But here you got anthracycline. So you got here actually five. So polar side chains and non-aromatic. So remember the polar chains are serine, thioiodine, asparagine, glutamine, and cysteine. Alright, that's how you remember them, because mostly of the OH group, you see the OH group here, you see the OH group here, you see other OH groups, and carboxylic groups as well. So yeah, polar side chain, asparagine, glutamine, cysteine, thioiodine, and serine. Alright, next we have the negatively charged acidic side chain. They have negative charges on the side group. So you have aspartate and glutamate. It's simple. The negative charge side group are two, aspartate and glutamate. Alright, you can see them. I don't know if you guys can see them that much, but anyways, aspartate, glutamate, two negative side chains. Alright, and they have the aspartic acid and the glutamic acid. So that's how you can remember their acids, or their acid forms basically. And lastly, you have the positively charged basic side chains. So the acidic negatively charged side chains are aspartic acid and glutamic acid. Next, you have the positive charged side chains. You got three. You got arginine, lysine, and histidine. Histidine has an aromatic ring, as you can see here. But other than that, you got three: arginine, lysine, histidine. Arginine, lysine, histidine. And yeah, this is basically just all the amino acids. 20 of them. They're gonna be really important when uncovering questions. Next, you have the amphoteric species, which accept or donate proton depending on pH. So an amphoteric species can act as both a base or an acid. So protonation, they gain protons under acidic conditions, and they deprotonate, they lose proton under basic conditions. Amino acids have a lot of proton in a solution, so they are very acidic and they fully pronate. Zwitter ion is when reaching a pKa of a molecule can have both a positive and a negative charge. So there comes a time when a certain pKa of a molecule can have both charges on the ion. So in order to calculate the isoelectric form of an amino acid, so it's the pK1 plus pK2, you're given two pKa's, and then you divide it by 2. If the pH is lower than the pKa, then the group will be pronated. It will gain protons under acidic conditions. Next we have peptides. Peptides are chains of amino acids. So amino acid is like the unit, and then peptides are chains of these units. Right? You have dipeptide, 2 amino acids, tripeptide, 3 amino acids, etc. But a polypeptide is the word you see the most. It means multiple amino acid chains. And they're joined together. The way the amino acids connect is through an amide bond. And the amide bond happens through condensation. A CN bond in the amide has a partial reaction. Next, that's when basically the protein structure or the protein synthesis happens. So it happens in um, the four different steps. So the first step, which is called primary protein structure, is the sequencing of the amino acid in the polypeptide chain, and you always start with the N terminus and end with the C terminus. So primary structure is sequencing of amino acids, start with N and end with C. Next, you have the secondary protein structure, which is a folded structure that forms the backbone of the protein. So you can have it as alpha helix or beta pleated sheets. So the alpha helix is carbonyl bonded to amino groups, R group, amino acids R group, and the beta pleated sheets are basically two or more segments of polypeptide that connect through hydrogen bonds. So basically this is like sorry for my drawing, this is the beta pleated sheet, you know, and then you got underneath it another sheet, and then they connect through what? Hydrogen bonding. But next we in the proline, remember proline if you can recall. All right, I want you to kind of recall what proline is. So proline was the non-polar, non-aromatic that occurred sorry, that we mentioned at the beginning. All right, and it's found between the beta pleated sheets and the residues of alpha helix. So proline helps with the secondary protein structure. Next we have the tertiary 3D structure, which the creation of like the groups, and the groups interact as 3D structure and occurs through ionic bonding, hydrogen bonding, dipole-dipole. So basically hydrophobic interaction and intramolecular bonds. Disulfide bonds contribute to the 3D shape between the cysteine side of the chain. Hydrophobic interactions occupy the interior of the protein because a salvation layer forms and hydrogen bonding cannot form with the amino acid side. Water molecules rearrange to increase entropy and decrease disorder. So basically the hydrophobic interaction occurs in the interior of the protein because there's a salvation layer that forms and hydrogen bonding cannot really form with amino acids, because for hydrogen bonding to form it needs to form with a H and then connected to a fluorine or a oxygen or nitrogen. So with the fact that this is not possible, that's why water molecules rearrange themselves for that reason. So thank you so much. Let me know if you have any questions, any inquiries I should answer. Don't forget to check our other videos, check our TikTok as well. Thank you so much.